I'm your next presenter, and I will speak about aphids in pulse crops and cereal crops. Okay, so we are monitoring aphids in pulse crops mainly for the last few years, and we are doing that work under the Grains Agronomy and Pathology Bilateral Program, which is actually founded jointly by investment of GRDC and New South Wales DPI. Why we are doing that? Well, because monitoring and early detection of aphids in different crops is key to effective management. Aphids can be very destructive crop pests, and they can damage plants directly through feeding damages, or they can cause indirect damages by secretion of honeydew, and also cause damages through the transmission of plant viruses. In order to support profitable cropping, there is a need to actually monitor different aphids and find the best way to understand their behavior, movement, and we are conducting this kind of work using different trapping methodologies, as you can see from this picture. Monitoring aphids is actually constant work. Environmental conditions can influence, which in 2017 was shown as being very unfavorable year for aphid and aphid multiplication. So we can see from this slide that aphid movement was very low in 2017. In 2018, movement was even lower, and which was reflecting environmental condition during the season. In generally, very low aphid movement was noticed with even with the mild autumn condition. So we could see that the aphid population was really low. So in some traps, even lower than 150 aphids per square meter per day. But even though we had such a low aphid movement, we noticed a very high population in our faba bean crop. So we found cowpea aphid build quickly in observed crops, and these dense colonies actually caused the damages together with the direct damages together with the production of honeydew. We noticed that in 2019 compared to 2017 and 18, it was still higher peak of aphids caught in our traps. Especially one peak was noticed from July to September. After that period, aphid number declined as crop matured earlier due to the extreme drought. At the same time, the population of beneficials increased, which you can see from next slide, where we actually find different beneficials in our crops, including lady beetles like on this picture, but also lacewings, hoverflies, parasitic wasps, and other predatory species. In 2020, we noticed that aphid numbers in our crops. Actually, we monitored the aphids in lucerne crop. We noticed that aphid population is actually at this moment is dropping down. So there were two main peaks observed depending on the environmental condition, and different aphid species were recorded. Among others, the main aphid species were cowpea aphid, spotted alfalfa aphid, cotton aphid, green peach aphid, and oat aphid. In this season, aphid infestation was reported in Mori district. During the inspection, actually high activity of beneficials was noticed together with the cowpea aphid. Even though the New South Wales thresholds for aphids in fava bean emphasize that there is need to treat very low aphid numbers in order to stop virus transmission, we considered not to use chemical control, as most insecticide would destroy beneficial insects along with the pests. Also, we expected the cooler temperatures would have significant impact on reducing aphid population, which actually they had. So we decided actually not to spray this crop. And one more fact that actually helped us to decide for such a thing, because after testing fava bean plants, there were no positives for presence of any viruses. Just to conclude the story on aphids in pulse crop, aphid population, generally aphid population dynamics are affected by environmental conditions, crop development, but also aphid biology and population of natural enemies. So please just consider that your decision on aphid control has to have integrated approach of all mentioned factors. One more thing I would like to point out, this is picture of Megura crassicauda, fava bean aphid, which has been found in parts of New South Wales, actually in Sydney, Tamworth and Breeza, in fava bean crop in 2017. It is large aphid with bright green body and dark head and legs. It has very intensive red eyes, as you can see from these pictures, which are very recognizable and easy actually to identify maybe this aphid. But do not bother with identification. If you find something similar, something suspicious looking like this aphid, please just contact New South Wales DPI or QDAF entomologist, and we will help you with the identification. So far in 2018 and 19, we didn't find this aphid, but we had 2 drought years, so it's possible that this year, this spring, this aphid could again be in our fava bean crop. Regarding aphids in cereal crops, different aphids could be observed. Oat aphid usually form colonies in lower leaves on base of plant. On the other hand, corn aphid and rose green aphid colonize the top of plants. Particularly, corn aphid colonize the whorl. Cereal aphids are known as vectors of barley yellow dwarf virus, and these aphids are found each year in crops in northern region. Russian weed aphid is something that you haven't been seen in your crops, maybe not all of you, but northern and Tamworth region, you haven't met Russian weed aphid yet. Hopefully you won't. Russian weed aphid was firstly identified in South Australia in 2016. In 2016-17, infestation stretched into Victoria and in southern New South Wales and Tasmania. In late 2018, first detections were made in north of Dubbo, then in Liverpool Plains and in Coonabarabran. To date, Tamworth, as I mentioned, is the most northern site where a Russian weed aphid has been confirmed. No new detections so far were made in 2020. Last confirmation were Bandella, Curabubula, Brisa, and Tamworth. But in 2020, we haven't even found them in sites where actually we confirmed its presence. So you shouldn't be afraid that this aphid will affect your crop this winter season. How to identify Russian wheat aphid? Russian wheat aphid is pale green, spindle-shaped aphid. Cornicles or siphunculas are very short, almost invisible. Russian wheat aphid has so-called double tail, which is actually formation of its caudal and supracaudal process, which actually form like two tails, and that definitely will help you to identify this aphid, because that is the feature that's actually that makes you make for you easy to distinguish it from all other aphid species. Russian wheat aphid host range is as follows. So firstly, barley is primary host plant, then wheat and durum wheat. Oats are resistant to Russian wheat aphid, and triticale is moderately resistant. There are no evidence of Russian wheat aphid being a pest in sorghum or in maize, but at Tamworth Institute, we are conducting research on level of potential damages in sorghum and role of sorghum that might play in maintaining Russian wheat aphid population over summer. As soon as we got results on these issues, we will definitely publish it. So far, we found Russian wheat aphid in our positives. It was from volunteer winter cereals and from barley grass. Russian wheat aphid can infest crops under warmer condition. Usually that's happening in autumn when it's mild autumn, or early in spring. Usually Russian wheat aphid causes problems when populations start to increase from tillering to stem elongation. Adults, so flights of adults occur in South Australia in October or during November, as the winter host starts to dry off. During summer, Russian wheat aphid has been found, as I mentioned, on different grasses, which provide green bridge for actually infestation of autumn plant cereals. Firstly, Russian wheat aphid will infest stressed areas in paddock, which you can see from this picture. So this is a little patch where Russian wheat aphid is actually found. Usually it forms colonies in patches and attacks younger leaves, which have to be unrolled to be inspected. Leaves infested with Russian wheat aphid have long white, usually white or yellowish stripes. Sometimes stripes can be purple. That's happening when plant has been exposed to lower temperatures. These symptoms appear because of saliva of this aphid, which is toxic to plant. So far, Russian wheat aphid in Australia is found to be manageable. So in recent years, conducted trials by Sardi reported that there is very little or no yield losses when Russian wheat aphid was controlled before GS40. So be aware that Russian wheat aphid is strongly regulated by environmental conditions. So, for example, in higher rainfall area, where, for example, we have like heavy rainfall. That's happening with Russian wheat aphid, that it's often washed off plant, and which cause actually 50% mortality of one colony. They prefer relative warmer, drier climates where annual rainfall is less than 400 millimeters. Existing economic thresholds for cereal aphids in general are more than 15 aphids per tiller on 50% of tillers if the expected yield will exceed 3 tons per hectare. But SARDI is conducting research on establishing thresholds suitable for Australian system. So until they finish their work, the US threshold, especially for Russian wheat aphid, needs to be applied, which is before GS32. If 20% of plants being infested, you should apply some insecticide, or after GS32 if 10% of tillers are being infested. If you need to apply any insecticide, please select the softest option, for example, Pirmicarb. Prophylactic foliar treatments are discouraged as they can affect beneficials. Also, natural enemies could limit the Russian wheat aphid population, which actually I witnessed in Bandela sites, and of course in Coonabarabran, where actually beneficials took under control Russian wheat aphid. If you find Russian wheat aphid in your crop, or if you are suspicious, you should follow this strategy. So firstly, look for symptoms. Be sure that it's not, for example, barley yellow dwarf virus or herbicide damage. Take a look, stressed areas firstly, unroll leaves so that you can see colony inside. And if you find colony that is still looking like Russian wheat aphid, or if you are just suspicious, just send photos of specimens or symptoms to New South Wales DPI or QDAS entomologist. We will inform relevant biosecurity entity. That concludes my presentation.